Hello, and welcome to an introduction to prokaryotes. Prokaryotes play a profoundly important role in the environment and on human health and other organisms as well. And so this video will just explore some of the basic biology of bacteria and archaea. Once you finish with this video, you should be able to describe the general features of prokaryotes. You should also be able to distinguish gram-positive from gram-negative bacteria. And you should also be able to explain how prokaryotes can actually move around their environment. So let's just begin with a few general features of prokaryotes. They're pretty much everywhere. You can go to the bottom of the ocean, to the top of Mount Everest, and you're bound to find various bacteria growing there. They tend to be quite small. For the most part, they tend to be much smaller than most eukaryotic cells. There are a few exceptions. If you look up on the screen, you see a fruit fly, and that white structure that you see, that's actually a single bacterial cell. So this is called Thiomargarita. It's a giant among prokaryotes. But again, for the most part, most prokaryotes are indeed much smaller than that. They also tend to be single-celled organisms, and most of them also have cell walls. We'll explore cell walls in just a few moments. But perhaps the feature that maybe comes to mind to begin with among bacteria is the fact that they tend to lack true membrane-bound organelles. So they don't have a nucleus, for example. They also don't have Golgi bodies, endoplasmic reticulum, or again, any other membrane-bound organelles. If you look at all the groups of prokaryotes that exist, all the different species, we can distinguish two separate and distinct domains of prokaryotes, namely the so-called bacteria or true bacteria and the archaebacteria or archaea. If you were to look at these two under the microscope, they would probably look identical. You really couldn't probably tell them apart from each other, but there are enough molecular differences between these two groups of prokaryotes that allows us to distinguish them as two separate and distinct domains. So just to get your bearing on this large tree of life that we see here, we've got bacteria, archaea, and then everything else, all the plants, animals, fungi, and protists, those are all included in this domain here, the eukarya or eukaryotes. So that just gives you some context for these two large groups or large domains of prokaryotes. If we look at the basic shape and structure of bacteria, we find some common patterns here. There tend to be fundamental shapes that are common to many species of bacteria. So, for example, toward the top, we see these spherical shaped cells. We refer to those as cocci. Toward the middle, we see these rectangular, box-shaped, sometimes described as rod-shaped cells. Those are bacilli. And then toward the bottom, we see these spiral-shaped cells, referred to as spirali. So in addition to these common shapes, we often find bacteria in distinct arrangements when we look at them under the microscope. So two of the common arrangements are the so-called strep and staph arrangements. So first of all, strep. Well, you have undoubtedly heard of strep throat. Often strep throat is caused by Streptococcus pyogenes. So strep refers to an arrangement of cells in a sort of chain-like manner. So if we take a look at this middle image, we see these cells are sort of arranged in this chain-like fashion. Well, that's the strep arrangement. Just another view shown schematically. Here we see some spherical shaped cells. So these would be streptococci. Again, this chain-like arrangement to those cells. On the other hand, the staph arrangement, you've undoubtedly heard of staph infections, often caused by Staphylococcus aureus. The staph refers to the arrangement of cells under the microscope, often occurring in a cluster-like pattern like we see here. So again, the point is often the shape and arrangement of cells is reflected in the various names of bacterial species. We mentioned a while ago that bacteria tend to have cell walls. So let's explore the basic structure of bacterial cell walls. I would point out that there's a very simple staining technique called the Gram stain, after the person who worked this out to begin with. that can allow us to help begin to identify an unknown bacterium. So let's see if somebody has a bacterial infection. If we want to find out what's causing that infection, one of the simplest things we could do to begin to identify the bacterium is a Gram stain. So it's a very simple technique. Once you do it, your bacteria will have either a purple color, like we see here, those would be gram-positive bacteria, or a red color, like we see here, those would be gram-negative bacteria. So what's the difference between gram-positive and gram-negative bacteria? Well, it comes down to their cell wall structures. So let's first of all take a look at gram-positive bacteria. It turns out gram-positive bacteria have a cell wall composed largely of this carbohydrate called peptidoglycan. So just to get your bearing, here's the cell membrane, and then just outside of that is this relatively thick layer of peptidoglycan. So again, that's the basic structure of gram-positive bacteria. On the other hand, gram-negative bacteria, slightly different structure. So toward the right, we see cell wall of a gram-negative bacterium. So again, here's the cell membrane, very thin layer of peptidoglycan, and then just exterior to that is yet another membrane. So again, the structural difference between gram-positives and negatives, that's what gives them the different coloration when you want to run a Gram stain on a sample. And this is important to know because if a person is indeed infected with some bacteria, some antibiotics work better on gram-positive bacteria, while others work better on gram-negative bacteria. It turns out that many bacteria actually have the ability to move around their environment. So how do they do that? Well, here are just a few examples. What we see up on the screen here is a single bacterial cell, kind of a corkscrew shape to it. That's called a spirochete. And it turns out this spirochete has these so-called axial filaments. These are proteins that run the length of the cell, and they can contract. When they do, that causes that whole entire cell to sort of corkscrew its way through its environment. So some spirochetes cause disease, such as syphilis or Lyme disease, and this ability to kind of corkscrew their way through their environment allows them to also burrow into host tissues. So that's just one way that some bacteria can move around their environment. Some bacteria also have flagella. But unlike flagella in eukaryotes, that if you think of sperm, for example, those flagella sort of whip back and forth. Flagella in prokaryotes works by a profoundly different mechanism. It turns out this may be the only example of a spinning wheel in living organisms. So here we see a sketch of bacterial flagella. Again, just to get your bearing, here's the cell membrane, here's the cell wall, but here's the flagellum protruding from the outer surface of the bacterium. And again, this whole entire structure can spin. So it works by a different mechanism completely than the flagella in eukaryotes. Some bacteria, for example, certain cyanobacteria or blue-green algae, like we see here, display the so-called gliding motility. They don't have any flagella or cilia or any real external structures that they use for movement, but they can use proteins in their outer surface of the cells to actually glide across various surfaces. So that's a very unusual type of motility, and we're still trying to figure out exactly how that works. In terms of reproduction, just a few comments here. Bacteria don't have linear chromosomes like we do, but of course they do have DNA, they do have a chromosome. It's sort of circular in nature. So they don't really undergo mitosis, but they undergo something comparable, referred to as binary fission. And that just simply indicates that their chromosome replicates, and then at that point, then your whole entire cell will divide into two separate cells. So sort of like mitosis, but again, they don't have chromosomes like we do. But bottom line is, through binary fission, you simply produce genetically identical copies right, of the cell that you're starting with. Nevertheless, there are various ways that DNA can be exchanged between adjacent bacterial cells. So this process of conjugation, that's sort of illustrated up on the top figure here. You can have, for example, a plasmid that can be copied and one copy given to another bacterial cell. That's what we see here. You can even have portions of the main chromosome that become copied and again given to another bacterial cell. So the point is there are ways we can have exchange of DNA between adjacent bacterial cells. Toward the bottom of the screen, we see an image of a bacterium, and this large structure here is an endospore. It turns out some bacteria are spore-forming bacteria, and these endospores, they form when conditions get rough. So if a bacteria is growing, but it runs out of water or nutrients, what certain species can do is essentially wall off their chromosomes, so wall off their DNA, and the whole entire cell will potentially die. But this endospore can survive. It's a very resistant structure. It can go long periods of time without water or nutrients, and then in the hopes that some point in the future it will encounter um, conditions that are favorable to growth, and it can then once again start to grow and produce an entirely new colony of bacteria. So just a few concluding comments. It turns out that bacteria play a profoundly important role in the nitrogen cycle. So if you think about it, we need nitrogen for various reasons. For example, think about DNA. Think about the nitrogenous bases, right? A's, T's, G's, and C's. So we need nitrogen. All organisms need nitrogen for various reasons. Most of the nitrogen that's out there is in the form of N2, gaseous nitrogen. Every breath we take in is composed largely of nitrogen, but the problem is we can't use that directly. It has to be converted to other forms, ammonium ions, nitrites, nitrates, and so forth, various forms that can be taken up directly by organisms. And we rely on bacteria and archaea to accomplish just that. And then once the nitrogen kind of makes its way through ecosystems, then once again, we rely on bacteria and archaea to put that nitrogen back into the atmosphere in the form of N2. So again, nitrogen fixation and denitrification is responsible, it comes about as a result of the activity of bacteria and archaea. So a profoundly important role in the ecosystem. And then lastly, it's important to recognize this large body of research that has become quite active in the past several years dealing with microbiomes. So if you look at the collection of different bacterial species that live in various parts of our bodies, and including our gut, it turns out that can have a huge role on a person's physical and mental health. So again, this is a very large and active area of research. So that was brief, but that concludes our introduction to bacteria and archaea.